Dear students, let me say several words about the basic histological techniques. This is one of two lectures about techniques, so not only cells, tissues, organs and their development are topics of the histology and embryology, but also I would like you know something about the methods, about the procedures, how histological slides are prepared, how do we work with the tissue. You already know that we have to use light or electron microscopes in histology and embryology because our eye has got very small resolution power. The resolution power of eye is about 100 micrometers. Everything what is smaller is in fact invisible by the eye without any equipment, and so we must use some type of magnification. A light microscope can increase our resolution power approximately 500 times, and the resolution power of a light microscope is thus 200 nanometers. 200 nanometers is quite good resolution because it can allow to see all cells which are usually bigger than 1 micrometer. There are some exceptional bacterial cells which have this size, but all human cells are bigger than 1 micrometer, and it can also allow to see some organelles which are bigger than 200 nanometers, like mitochondria, some lysosomes or granules. They are not organelles but inclusions. Nevertheless, they can also belong to group of structures which are visible by light microscope. But there are many smaller things, many things, many structures in cells and tissues which are below the resolution power of the light microscope, like ribosomes or cytoskeleton, or molecules, macromolecules, nucleic acids, proteins. All these structures are visible only by the electron microscope, which is increasing our resolution power more than 1000 times to 0.1 nanometer. This is a resolution power which can allow to see almost all structures of a cell. Of course, not atoms. Atoms or small molecules are below this level, but large molecules and their combinations are visible. So the reason why to increase is clear. Now let's look at several techniques which can allow to see the increased picture. The methods for their preparing are very similar in both light and electron microscopy. Light microscopy and electron microscopy are two disciplines with very similar technical base. In both techniques, the first step has to be sampling. The second step has to be fixation. I explain what does it mean. The third and other steps are usually, not every time, that's why are there these parentheses, but usually dehydration, clearing and embedding. Necessary for transmission light and electron microscopy, the necessary procedure is sectioning, because both these techniques of microscope are based on the transmission of light or electron beam through the very thin specimen. The specimen, the slide has to be very thin. In light microscopy, usually in micrometers. We can measure its thickness in micrometers. In electron microscopy, the specimen has to be thinner, about in tens nanometers. So sectioning, you can see that sectioning is the last step, which is really common for light and electron microscopy. Then procedure is a little bit different, and it's possible to explain very simply. The light microscopy is based on the visible light usually, and it means we can use colors in this method. And to use colors better than with natural colors of the specimen, we should stain. The electron microscopy is based on the transmission of beam of electrons, and colors are not usable here. But the picture from the electron microscope can have a variable contrast, and so to improve the contrast of the picture, there is contrasting instead of staining. So staining, contrasting are in fact very similar methods, improving the visibility and the resolution of the picture. The slides have to be mounted also. Mounting is in both techniques, but little different moments before contrasting but after staining. And the last step is of course observation. Without observation, the whole previous work would be fruitless. So all slides which are prepared in the laboratory have to be observed. We are beginning with sampling. Sampling of a specimen can be from living organisms. It is so-called biopsy. You probably know this work. And this biopsy is mostly diagnostic. Diagnostic biopsies are made in surgeries and other departments, and these specimens are then sent to pathological and histological laboratories, then processed and evaluated to say something about the diagnosis of our patient. Biopsy can be also experimental. There are experimentals with laboratory animals where we also collect specimens. The method how to obtain some bioptic material can be very variable. Probatory excision means just sectioning of some small piece of tissue. Needle biopsy or so-called punction, you probably know that liver or kidney or thyroid gland can be evaluated by this method in pathological laboratories. Curettage is a method which is used usually in gynecology, microrelief. All these methods can be used by surgeon or gynecologists or other doctor to obtain the tissue. Then the tissue is sent to lab. Necropsy is the sampling of dead bodies, of course. It can be also diagnostic. Maybe it's surprising for you, but we like to say more about the diagnosis of our patient even after his or her death. There can be supportion of the treatment procedure, or this after that necroptic diagnosis can say something against the previous procedures, and it can be new education for the doctors. So diagnostic necropsy exists. necropsy can be also experimental from laboratory animals, and necropsy is legal also for teaching. So our slides, which you can find in the boxes in our study rooms, our microscopic rooms, are usually from necropsies which were collected for teaching, because we ask our colleagues in pathological departments for some tissues, organ we need. After the sampling, the tissue has to be fixed. Why? Why? It is important to stop all vital processes in the tissue. Vital processes, what does it mean? It means mainly enzymatic processes. Almost all biological procedures, metabolic procedures are catalyzed by enzymes, and the stop of these enzymes means also usually the stop of the vital processes. So the principle of fixation is to stop the enzymes, and as the enzymes are usually proteins, the universal method how to fix and so stop enzymatic functions is a coagulation of tissue protein. Coagulation of tissue proteins using chemical substances or physical agents is a universal method of fixation. After that, the tissue doesn't change. It's so-called autolytic, I think I can write this word, autolysis. Autolysis is a process of the enzymatic changes of tissue after its death, and it starts immediately after the decrease of oxygen levels in the tissue, and it's go on with, up to this moment when we fix the tissue. Autolytic changes can sometimes damage the tissue so much that it is impossible to recognize anything there. So that's why we'll try to stop it as early as possible. Because if we do it, we can preserve specimen structure. That's the thing which we need. It also preserves staining abilities. It's also necessary for histology. Important is a penetration speed. Some fixations, some fixatives are penetrating quickly to the tissue. Some are smaller. So that's the thing the laboratory staff has to know to adjust the size of the specimen and so on. What are the fixation methods? As I already said, they can be physical or chemical. Physical methods are in fact quite simple. It's mostly use of heat, either low or low temperature or high temperature. More frequently is low temperature, and that means freezing. Freezing is a fixation which you also know from home. If you have a food which is necessary to store, you can freeze it to prevent biological processes in the tissue or in the vegetable or so. And so the freezing in the kitchen, in the refrigerator, is in fact fixation. In histology, we have to fix much faster and deeper, because simple freezing in the refrigerator is so small that crystals of ice are developing in the tissue, and these crystals of ice aren't a problem in kitchen, but they can be problem in histology, because they are very sharp and they can damage the cells and tissue. So we must prevent formation of crystals, and the best prevention is very fast and very deep freezing by, for example, liquid nitrogen, which has a temperature of about 200 degrees minus under zero. If we not only freeze the tissue but also dry, it is so-called lyophilization. It's also a fixation method. And heating, boiling, it's a method from kitchen again, and it's used also in microbiology, where bacteria have got very strong walls, but it isn't possible to use it in histology of human tissues. But freezing, yes, freezing is used. I'll say something about it in this lecture and more in the next lecture. There will be second lecture. Probably I already said it. There will be a second lecture in the second semester about special techniques. So I will not say everything today about the histological techniques, but I will say enough for the first test, for example, and for the basic knowledge about the tissue, about the histological procedures. The second and much more used fixation method is chemical. We can use vapors, but not very frequently. Much more often we use solutions of chemical substances, which can be very variable. We can use them by two ways: immersion and perfusion. What does it mean? Immersion means that we take a bottle with some fixative fluid, fixation, and we'll immerse our tissue into it, so then the tissue is flowing in the piece of tissue is flowing in the bottle, and fixation penetrates into it from the surface to the center, and its penetration is dependent on the penetration velocity. It's very important, that's why it's very important to know it. Perfusion is much more sophisticated method of fixation, which has a big advantage that the tissue block, tissue piece is fixed all together. There aren't any differences between fixation of surface and central part of the specimen, because it's based on the injection of the solution into the vascular system of our specimen. So we have to find a vessel which is going into our specimen, either vein or artery, more frequently artery probably, and we very slowly and gently perfuse the specimen by the fixation, by the fixative solution. The fixative solution slowly penetrates to the tissue by its own vessels and is diffusing through the capillary wall directly into tissue. Capillaries are almost everywhere, and so the whole tissue is fixed in one moment. Something about the freezing. Freezing, as I already said, has to be very fast and deep, so liquid nitrogen or other chemicals are used. Then the tissue has to be also stored in very low temperature. Special containers with liquid nitrogen can be used for the storage. There exist storage bottles, but also there are deep freezing boxes in the laboratories where minus 80 degrees of Celsius are maintained. It's much more, or in fact much less, much lower temperature than in a normal kitchen refrigerator, which varies usually about 20 18 to 20, 22 degrees, not less. Freezing can be sometimes combined with chemical fixation, and in this case we are using cryoprotective agents. Cryoprotective agents can be added to the fixation, to the chemical fixation, or are used after the chemical fixation to protect the tissue against formation of the ice crystals. So, for example, if we haven't got liquid nitrogen or some another solution necessary for the fast and deep freezing, we can overcome this problem by using of cryoprotective agents. Sucrose is the most frequently used of them, and this tissue which is infiltrated by sucrose can be then frozen normally in the fridge, and crystals don't develop, or maybe only small crystals are developing there. Chemical fixation. As you can see, there are very many fixation agents, fixation solutions, or fixatives. But I would point the first group, aldehydes. Aldehydes are really worldwide most used fixatives, and among them formaldehyde is the most frequent and probably the best. Formaldehyde is used in 4% water solution, which is called in histological laboratory language 10% formal, because formaldehyde is bought as a 40% solution, not pure, and so if we solve this, if we dilute this 40% solution 10 times, we'll get 10% formal or 4% formaldehyde. Formaldehyde is used mostly for light microscopy, but in some mixtures also for the electron microscopy. But the favorite aldehyde in electron microscopy is this another one, which is called glutaraldehyde. Glutaraldehyde is more complicated molecule than very simple formaldehyde. It penetrates a little bit slower into the tissue. That's why the specimens for the electron microscopy has to be very small. But when penetrating, it fixes the tissue very well without formation of artifacts like in formalin. In the electron microscopy, we use it usually as the first step of two-step fixation. In the electron microscopy, we usually use two-step fixation, and the first step is called pre-fixation, the second step post-fixation. So in electron microscopy, the glutaraldehyde is used for pre-fixation. There exist many other chemicals used for fixation. I think it isn't necessary to speak about all of them. Maybe I can mention osmic acid or osmium tetroxide, also soluted in water, which is used as the second step of fixation in the electron microscopy, so as a so-called post-fixation. Osmic acid or osmium is a heavy metal, and you already know that contrast in electron microscopy is based on the bond of heavy metal atoms on the surface of our tissue. To bind osmium into other specimen, we can use this osmic acid during fixation, and that's why this step is also called pre-contrasting. So the first step of contrasting of the tissue is done already during its fixation. And then there exist many other acids, salts, alcohols. Look at them, read them in the presentation, but I think it's not necessary to speak more about them. So let's remember that aldehydes are the most important chemical fixatives. After the fixation, which can last some time, which depends on the velocity of penetration and on the size of the specimen, so that's why we have to know it. The tissue has to be trimmed. Trimming of the tissue is a very important step, because only after this procedure we can orient the specimens in blocks. We can say to our laboratory assistants how do we want to orient the tissue, if we want to section it longitudinally, transversely, circularly, if we want to trim and cut everything or only some parts of our specimen, and so on. After the trimming, we have to wash our tissue, because the fixation agent would interfere with other steps, so we have to wash it from the tissue. Washing is very simple usually, because we use tap water for washing. But without this step, our next procedures wouldn't be successful. So never forget to wash the tissue very well. After washing, we can start the procedure of embedding. And the first step in this quite complicated procedure is dehydration. Why? Because the media which are used for the embedding of tissue, and embedding is necessary step to cut it. We are usually not able to cut the tissue without embedding, because it's usually very soft. So the embedding of the tissue is necessary, and media which are used there are hydrophobic. They don't mix with water. Usually, there are some exceptions, but the most frequently used media like paraffin or epoxy resins, celloidine, are not mixing with water. So we have to extract all water molecules from our specimen. It must be done gradually, slowly, because if we do it too quickly, the tissue can shrink, and the histological specimens would be deformed. So slow, gradual extraction of water from the specimen is very important. It's done usually with using alcohol or acetone. These solutions are given to our tissue in so-called graded series of concentration. It means we'll start from low concentration of alcohol or acetone, and then we gradually transport our tissue into more and more concentrated solution, and the final solution are 100% concentrated, so there is no water in the solution and all water from our specimen is extracted. But it's not everything, because the paraffin or epoxy resins or other embedding media are not solvable with alcohol or acetone as well. So the replacement of water by alcohol or acetone doesn't solve the problem. The tissue, the next step has to be to extract this dehydration medium from the tissue by a dissolvent of the embedding medium, and this is called clearing, because the tissue becomes usually translucent during this procedure. The dissolvent of paraffin is some aromatic hydrocarbon. Mostly used is xylene. Benzene was also used, but it is carcinogenic, so it was forbidden. Now xylene is used as a clearing medium in the paraffin technique before embedding in paraffin, and the most important embedding medium in electron microscopy is an epoxy resin, and its dissolvent is propylene oxide. So propylene oxide is another important clearing medium used for electron microscopy. So after dehydration, there are several baths of the tissue in clearing medium. Now we have got our tissue dehydrated and clear, and we can start with proper embedding. The first step is infiltration, because we have got now the embedding medium dissolvent in the tissue and the clearing dissolvent in the tissue, and we must replace it. So the histological procedure before embedding is still replacement of some solution by another solution. The last of these steps is infiltration. The tissue is infiltrated by melted paraffin or liquid resin. This proper embedding is submersion of the infiltrated specimen to the embedding medium. Paraffin is melted in higher temperature, slightly below 60 degrees Celsius. The epoxy resin or other resins used for the electron microscopy are usually liquid in the room temperature. So our specimen is submerged or immersed into this solution and then hardened. Paraffin block is hardened very simply by the cooling in the room temperature. Resins are usually polymerized in special ovens by high temperature or under UV light. As I already said, the most important embedding medium for light microscopy is paraffin. Paraffin is a wax, you know, as a material for candles, for example. So paraffin is widely used solution or widely used material. It's water insoluble, and maybe 99% of tissues in histological laboratories worldwide are embedded into paraffin. For light microscopy can be also used harder material than paraffin, which is called celloidine. It's good for embedding of hard tissues, like bone, for example. And there exist also water-soluble media which are used in some special circumstances. For electron microscopy, or maybe better is to say for transmission electron microscopy, because there exists also scanning electron microscopy. I will speak about it at the end of the lecture, which doesn't need any embedding, but for the transmission electron microscopy there are some resins. Resins are harder than paraffin because we need very thin sections, very thin slides, and the harder is the embedding material, the thinner and the more gentle can be sectioning. So please remember this simple rule, and that's why we use resins in the electron microscopy. Mostly epoxy resins, not so much at current histology polyester resins, but well used are acrylic resins because they are at least in part miscible with water, and so the tissue is not necessarily completely dehydrated, which can be sometimes good for some special techniques. Here is the slide showing all steps of the paraffin embedding. Dehydration. In paraffin techniques we use alcohol. We'll start by 70% alcohol and we finish by several baths in so-called absolute alcohol. Then clearing mostly in xylene, which is universal dissolvent of paraffin. Then paraffin infiltration, usually 3 baths, and proper submersion of the specimen with melted paraffin. The same, of course, we can do it by hand, but the bigger laboratories have got some machines, some histological techniques. This tool is called tissue processor, and it allows automatic dehydration, clearing, and infiltration of the tissue. Only the proper embedding has to be by another machine, which is called embedding center, where we can use some molds of variable size according to the size of our tissue which is infiltrated by paraffin is given here, is given to this mold, and from this thing the paraffin is going down and fills the mold, and the result is a tissue block, which is then cooled by cooling plate. Of course, it must be also labeled by a number of the specimen, and the result after the total cooling and so hardening is a paraffin block, where we can see some pieces of tissue in the big block of the wax. Molds can be metal or plastic, it depends on the laboratory. Celloidine, that's, as I said already, it's harder material for light microscopic embedding, used for hard tissues. Dehydration is very similar here as in paraffin technique, but clearing is different. There isn't any aromatic hydrocarbon, but ethanol and ether in mixture. Then the tissue is infiltrated directly by celloidine, embedded and hardened. The hardening of the celloidine can be done in the room temperature, and it's quite complicated. We have to use evaporation of ethanol ether and then ethanol bath. The result is harder block, can be similar in size but can be harder than the paraffin block you saw on the previous slide. Water-soluble media, as I already said, are used in some special situations. Gelatin or celloidal. I think it isn't necessary to speak about them more. In the electron microscopy, resins are used every time the tissue has to be dehydrated. If we use acrylic resins, this dehydration needn't be complete. So in these techniques we don't use absolute alcohol. During dehydration we can use the solution of uranyl acetate instead of water. Why should we do it? Uranium is another heavy metal which is used for contrasting, and we can use the step of dehydration for the next pre-contrasting. The first pre-contrasting was done by osmium during fixation, and uranium can be added to the tissue during dehydration. We are trying to improve the contrast of our specimens in every step of this procedure. Then it's clearing if necessary, infiltration and embedding. Instead of metal or plastic molds we use for the paraffin or celloidine blocks, here in electron microscopy are used smaller molds which are usually made from gelatin capsules. Gelatin is then dissolved and we have got, after hardening, very hard tissue block. Tissue in this block is usually just very small piece, about 1-2 cubic millimeter in size, because during the procedure we use high molecular fixatives like glutaraldehyde or osmic acid, and the penetration of these solutions is not so fast as the penetration of very simple formaldehyde, for example. Again, some laboratories where higher amount of resin specimens is prepared can use special tissue processors for many specimens which are processed together. The next step, when we have a block, either paraffin or celloidine or resin, is a sectioning. Sectioning is universal for light and electron transmission microscopy. We need, in all these techniques, we need thin sections. Sectioning of paraffin or celloidine or water media embedded blocks is done in the machine which is called microtome. If we want to section some resin blocks, we use ultramicrotome. What is the biggest difference? The biggest difference is the material of knives. Microtome is using steel knives or blades, ultramicrotome has to use much harder material, because the harder is material, the thinner sections can be done, as I already said, or can be made. So the hardest material at all is diamond, and really diamond knives are made and are used for electron microscopy sectioning. But glass is also hard enough, so instead of diamond, also glass knives can be used. And special machines for the sectioning of frozen specimens are developed as well. They use usually steel knives or blades, the same as are used in microtome, but they are especially equipped by freezing boxes to maintain the low temperature all the time, and they are called cryostat or freezing microtome. So let's look at these tools. Sliding microtome is the most important and most frequently used microtome for paraffin or celloidine sectioning. This is the picture from our lab. This microtome has got here a blade or knife can be here. It's steel, and here in this part of the microtome the paraffin block is fixed, and the laboratory assistant is moving by this part of the microtome forwards and back, and during each way of the knife along the block the thin section is made and can be then collected. So that's this movement forward and back is the reason why the microtome is called sliding. Another microtome is a rotary microtome, where the laboratory assistant doesn't slide the knife, but she or he rotates this handle, and the tissue block, which is here in this case, is moving along the blade situated in this red holder. Both these microtomes are used worldwide in laboratories. We have got several sliding microtomes. Unfortunately, we haven't got a rotary microtome. We want it, but it's a little bit expensive this time, so we have only sliding microtomes. Here is a detail of the work in a sliding microtome. You can see a tissue block here. Some tissue is inside, I don't know what is it, but some organ probably, some piece of organ. The laboratory assistant made the sliding movement of this blade, it is the blade here, along the surface of our block, and the section is here. This section can be then collected from the knife and put on the glass where a drop of water is given before. Now this thin section is floating on the surface of the drop. The thickness of this section is usually 3 to 5 micrometers. Light microscopy can use micrometric thickness of the specimen, so 3 to 5 micrometers is good. Sometimes some tissues which are difficult for section need to be sectioned to thicker section, 10 micrometers maybe, but never more. So 3 to 5 micrometers is optimum. The water droplet is then dried, evaporated, and as you can see here, our section is now situated on the glass, and that's the thing which we want. Of course, it isn't the end of the procedure. We now have a tissue on glass, but it is colorless, with exception of some natural colors like pigments, and it is still embedded in paraffins, and this paraffin here everywhere around, and not only around but also in the tissue, is a material which now has to be removed, because when we want to stain our section, we have to remove the paraffin as a hydrophobic material, because the staining dyes are every times water solution, sometimes alcohol solution, but they never mix with paraffin. The section wouldn't adhere to the glass automatically. We usually have to help with adhesion. There are some materials which help adhesion, like glycerol and equite. Its mixture is given to glass before the section is prepared, and it helps to adhere. Sometimes equite isn't possible to use because it contains proteins, and if we want to do some immunohistochemistry, it can be problem to have some chicken protein there. So another materials are used for improvement of adhesion, like silan or polyl-lysine. If we want to prepare our sections from resin blocks, we must use more sophisticated machine which is called ultramicrotome. Ultramicrotome is using sharper knives, is using another holders of specimen, because the specimen is very small, and as everything is there small, not only specimen but also sections, the ultramicrotome is equipped by binocular eyepiece to increase the scena here in the area where is sectioning. This blue thing is a knife, and this white thing here is a specimen, is a block, resin block. The work with ultramicrotome is usually automatic, so there aren't any sliding or rotation movements. Everything is moving automatically. There is a panel where we can set up all the properties. To section in the ultramicrotome, we need knives, and as I already said, they can be diamond, which can be bought, so there isn't anything difficult to prepare them, that it only needs money. And there is also possible to use glass knives, which are usually made directly in a laboratory by a simple tool now called knife maker. So there exists a knife maker which is breaking long strips of glass into squares, and these squares are then broken once more diagonally into two triangles, and this triangle is a knife. Its edge is very sharp, and some plastic well is is added to this end where the sharp edge is to be able to use water. Also here is a mold or well for water, because the sections are so small that the manipulation with them in dry space is impossible. They have to flow on the water level. Here is a detail of the knife and specimen in the ultramicrotome. Here is a detail of the panel of the directing panel of the electron microtome, of the ultramicrotome. And the most important value which we have to set during the sectioning is the section thickness. Section thickness in electron microscopy has to be between 50 and 100 nanometers. So we use, for example, 70 nanometer thickness, 90 nanometer. Everything what is thicker than 100 nanometers, like this or this thickness, is impossible for the electron microscopy itself, but it's still good for light microscopy, and it is really used for microscopy. The resin blocks are not only for are not only for electron microscopy, they can be used for both these types of microscopies. The sections which are for electron microscopy, so which are between 50 and 100, I lost my pen, yeah, here. These which are between 50 and 100 nanometers are called ultra-thin sections. The thicker sections are called semi-thin sections. So there are ultra-thin and semi-thin sections. Semi-thin sections can be used in light microscopy, and why do we use them is here written. We use them for orientation and selection of a specimen which is suitable for ultra-thin sectioning. So let's imagine the situation. We have got several tissue blocks from some patient or laboratory animal or something like this, and we must to select one or two of them for the gentle ultra-thin sectioning. How to select it? It's very simple. We prepare several thicker semi-thin sections. We'll quickly look at them in glass after short staining. So this specimen can be prepared during 1 hour. We can look at it and we can see, do I want it? Do I want to observe by the electron microscope this structure which is here visible, or no? If yes, the laboratory assistant can go on the sectioning and ultra-thin sections can be prepared. If not, another block is taken. Again, semi-thin sections are prepared at first, and then the scientist or the physician is deciding whether this is suitable or not. When you look at the sectioning in the ultramicrotome, you can see sections on the water level. And they have very variable colors because the light is penetrating through them or reflecting from them, deflecting, and so we can distinguish the thickness of the sections according to their color. For the transmission electron microscopy, the best are silver or gold sections, which are between, let's say, 60 and 100 nanometers. Everything what is violet, blue or green, red is too thick, but can be used as a semi-thin section in the light microscopy. So we recognize the thickness of the section according to its color. Ultra-thin sections are mounted instead of glass into the metal grids. So glass is a material which is impossible in electron microscopy because electron beam doesn't go through the glass. It would completely reflect. So metal grids are used. Metal grids are small circular pieces of metals, usually cuprum, sometimes gold, nickel. You can see that their structure, their raster can be very variable, and they can be used without any other coating, so-called naked grids, but they can be also coated. They can be coated by support film to improve the adhesion adhesion of tissue to the material of the grid, to the metal. It is like adhesion substances in light microscopy which are given on the glass. Several words about the sectioning of frozen specimen. Frozen specimen can be sectioned by cryostat, which is combination of rotary microtome and freezing box. So rotary microtome you recognize according to this wheel with a handle. So the laboratory assistant is turning this wheel, and microtome inside is sectioning. You can look at it through glass because in this chamber here is a low temperature to maintain our tissue frozen also during sectioning. Or you can use freezing microtome instead of cryostat. If you use fixed tissue which wasn't frozen before sectioning, it was chemically fixed, but now we need to section it to make it harder and to allow the formation of sections, of thin slides. The freezing microtome has to be quickly cooled before the sectioning, and it is possible usually by carbon dioxide expansion, but also so-called electrothermal cell or compressor freezer can be used for this cooling or freezing of the freezing microtome. The principle is similar as the sliding microtome. Here should be a knife in this place, and here is a specimen, frozen specimen, and movement of this handle here in this direction leads to formation of a section. Why do we use cryostats or freezing microtomes and the whole freezing techniques or cryotechniques also? The most important reason is that these methods are very fast. To freeze a specimen, it's a question of some seconds, and the sectioning can last several minutes if there is the good staff with good sectioning skills. And so the result of the histological examination can be known during, let's say, 1 hour if we used freezing techniques. When this quick procedure is necessary. It is necessary when we want to know the result very quickly, and it is the situation of rapid intraoperative biopsies. Rapid intraoperative biopsies are made by surgeon during operation, usually during operation of some tumors. The surgeon doesn't know, because macroscopically it is impossible to judge whether the tissue which is operating is still tumor tissue or if it is healthy tissue, but rapid biopsy can help to decide and can help to say whether the size, the level of this operation was sufficient, or whether there is necessary to do bigger operation and bigger segment of some organ has to be taken. This rapid intraoperative biopsy is done during the operation. The patient is still on the operation theater, is still under the general anesthesia, and the whole team is waiting for the result of the histological examination, and that's why it must be very rapid, very quick. The other reasons of freezing techniques are not the velocity of this examination, but the fact that some structures, some molecules, some substances are dissolved during paraffin or resin techniques, like lipids. You already know probably that lipid droplets are dissolved during paraffin techniques, and if we want see lipids and if we want stain lipids inside, we have to use freezing methods, and there are some another reasons of the freezing. So freezing is used, and that's why special microtomes for freezing specimens were developed, like cryostat and freezing microtomes. And now we are at the end of the part of my lecture when everything was almost common for light and electron microscopy. It was possible to speak about both microscopies together. There were just small differences, or sometimes procedures were the same, like dehydration can be really the same for light and electron microscopy, or another steps as well. Now staining is possible to use only in light microscopy. The reason was already said. Only light microscopy can show colors. Staining isn't only introduction of colors into the slide to make it nicer, but it is important for recognition of components, because it isn't interaction of the whole specimen with some color, but it is interaction between color and some certain tissue components. Color is also called dye in histology. So histologic dye is interacted, is bound during staining into certain tissue components. If we use combination of dyes in order to stain the whole specimen, the staining is called synoptic. If we use synoptic staining, we can see everything in some colors, or almost everything has some colors, and it is possible to distinguish the structure which were originally acid or basic, because the synoptic staining is mostly based on the acid-base affinity. Acid-base affinity is the crucial principle of the synoptic staining. That's why we must every time use at least one basic dye and at least one acidic dye. Usually, or not usually, the most frequent is hematoxin and eosin, which is using just one acidic and one basic dye. But some another synoptic methods are using more dyes, usually one basic but 2 or even 3 acidic dyes. You can see that there aren't only hematoxylin and eosin. Hematoxylin is blue and eosin red, and they are the main synoptic dyes, but you can read another names of dyes, and every time the name is written by the color the given dye has got. So you can see that not all basic dyes are blue or violet, that there exists also red basic dyes or also green. Methylene green is basic. And also acidic dyes are not only pink or red like erythrosine or eosin, but again also green, yellow, also blue, orange. So there is the whole spectrum of colors. So that was synoptic staining. Then there is a big group of special stainings. There are plenty of methods of stainings which are used when we want to highlight something specific. We are looking for something, we are interested in some special structure or a special substance in our specimen, so we use special staining to visualize and highlight it. So the special staining is based on the affinity of a special dye and some specific substance. At first, something more about synoptic stainings. Synoptic stainings, or maybe this slide is valid also for most special staining, the basic staining procedure. At first, we have a paraffin section on the glass. You already said them in one photograph. There was a section on the surface of a glass, but it was surrounded by paraffin, and we know that also inside the tissue is a paraffin because it was infiltrated before the sectioning or before the embedding. And so the first step has to be slight deparaffination. We have to remove the paraffin because it is hydrophobic. When we haven't got paraffin in the tissue, we use water to rehydrate it. So the second step is rehydration. Why? I think that I said already that the histologic dyes are water or sometimes alcohol solutions. Then can be the staining. To preserve the tissue, so the staining can be the step 3. To preserve the tissue, the slide should be once more dehydrated, because water is a good environment for some molds or yeasts and bacteria, and it can damage our specimen. So to conserve the specimen, it must be then once more dehydrated and then once more cleared before mounting. So dehydration, slight dehydration and slight clearing are in fact repetitions of the same steps which were done at the beginning of the whole procedure after fixation. In practice, we use baths of chemicals like alcohol, carboxylane, xylane. It is for clearing and dehydration after the staining. You can see that in this set the hematoxin and eosin was prepared. The tissue is on the glass, so this is the holder of the glass. Glasses are situated in this holder, and the laboratory assistant is moving this basket with glasses from one bath to another bath and then to another bath. Every time the time of the bath is also important, and at the end there is the stained specimen, stained and dehydrated and cleared specimen. Here is the recipe of the most frequently used staining method in light microscopy, hematoxin and eosin. So deparaffination and rehydration, here xylane and alcohol is used, then water at the end. Then staining can start. Usually we use at first basic dye and at second eosin, like the example of acidic dye. Washing has to be between all staining steps has to be washing. We already know that hematoxylene as a basic dye is binding into the acid components of the tissue, and these acid components are then called basophilic. The same is with eosin. Eosin is acid dye. It stains basic components of the tissue, mostly proteins, and they are then called acidophilic or eosinophilic. I think that everything you know. Then again alcohol is used for dehydration. Once more, xylane is used for clearing. You saw it in the previous photograph. And the last step is mounting. Mounting is covering of the slide with the cover slip. This is the picture of the specimen of esophagus, in this case, stained with hematoxin and eosin, so we can see red or pink structures which are stained by red color, red dye, which is eosin here, and we can see violet structures like here or here, which are usually nuclei of cells stained by hematoxylene, a representative of basic dyes. There is a table showing another synoptic methods of staining. So hematoxin eosin is the most widely used, but these other 5 methods are also widely used. You will see most of them in our slide box. Bigot-Fangison and Masson trichromes are used mostly to increase the difference between connective tissue and muscle tissue, to improve their recognition. Collagen is stained by one acidic dye, and muscle cell or muscle fiber cytoplasm is stained by another acidic dye, and so at least 2 acidic dye are here used in this staining. Like in Bigot-Fangison, it's acidic fuchsin and picric acid. Acidic fuchsin is red and stains collagen. Picric acid is yellow and stains muscle. And some similar combinations are in trichromes and in azan. Azan is the only of these 5 methods in which another basic dye is used. Instead of hematoxylene, there is azocarmine used, and that's why nuclei stained by azan method are red. In all other techniques, nuclei are stained with hematoxylin, which can be either brown to black if it's iron hematoxylin, or blue if it is normal hematoxylin used in hematoxylin and eosin staining. Okay, so please remember the other synoptic stainings: Weigert-Van-Gisson staining, three Massons, trichromes which differ in the staining of collagen, yellow, blue and green, and azan as another example of synoptic staining in which nuclei are red. Pictures of photographs from the specimens stained by these another synoptic staining. Weigert-Van-Gisson can show red connective tissue. These red structures here are collagen fibers, and yellow are bundles of smooth muscle cells here. It is again specimen of esophagus, but muscle in this case is yellow and connective tissue red, so it is very easy to distinguish them. When we look to previous picture of the esophagus stained with hematoxylin and eosin, we can see muscle here, which is red, and we can see connective tissue with collagen here, and you can see that it's also red or pink, maybe little lighter but not so much. So to distinguish connective and muscle tissue in hematoxylin and eosin is not so easy. It is not impossible. You will know it. You will be able to distinguish them. Don't worry. There is another structure. Muscle tissue is composed of cells. Each cell has a nucleus. Connective tissue is composed mostly of extracellular matrix. So there aren't nuclei in collagen fibers because they are not cells. So differences are here, but sometimes it isn't so easy as in Weigert-Van-Gisson or trichromes, where it's opposite. It is yellow trichrome and connective tissue here is yellow, and muscle, which is here, is pink in this case, or here is a smooth muscle, it's again pink. It isn't esophagus, it is uterine tube here, but it doesn't matter. Or blue trichrome, where muscle is red here and collagen is blue. Or green trichrome, when muscle is again red. It is red in all three trichromes, but collagen is green here. Also, muscle is red in all trichromes, but yellow in Weigert-Van-Gisson staining. Again, when the laboratory is so big that the automatization is necessary, there exist staining processors. And now we'll look at special staining shortly. As I already said, the number of methods used for special staining is enormous. There are plenty of methods, and my lecture can contain only small choice of them. The principle is to highlight some search structure, so this dye which is used here has to have some specific affinity. And what is very important, that during the staining there isn't any color chemical reaction. No color chemical reaction is here. If there is chemical reaction with the change of color in during the formation of slide, it isn't staining but histochemical reaction. I will speak about histochemistry next time in the special techniques. So staining is based on the principle that there is no change of color. The dye has its color. For example, dye is yellow and result is yellow. Dye is blue and the result is also blue. If dye is, or if this substance given to the specimen is colorless and result has some color, it isn't staining but histochemistry. As there are so many types of special staining, we have to classify the special stainings. There are 3 categories of special stainings, which are called cytological, selective, and impregnation staining. Cytological stainings is the staining of something in the cell, something in, sorry, here should be R, something intracellular. Selective staining is staining of something regardless of the location, so it can be something intra or extracellular, like some proteins of extracellular matrix, which can be also before secretion in the cell. And impregnation methods, they are special stainings based on the reduction of metals, mostly silver. Silver reduction, Ag is silver, so silver reduction on selected structures. I will show some examples. Cytological stainings can be very variable. Here are several of them named, but what would I emphasize? Maybe staining of Nisl substance. I will every time try to find some techniques, some special techniques which were used during the formation of slide box for teaching in our department. Our slide box doesn't contain only synoptic staining. Usually, yes, maybe 90% of specimens is hematoxin and eosin staining. Then there are several specimens stained by another synoptic methods, like Weigert-Van-Gisons or some trichromes. And then there are several specimens where special staining was used. And at the end there are some specimens from histochemical methods. Special staining is used in maybe 5 slides. I don't know it now exactly, and I will every time highlight this method which is used in our box. So there is one cytological staining in our slide box, and it is the staining of spinal cord by Nisl substance, or not by Nisl, it's the staining by toluidine blue, and it is the staining of the Nisl substance. Nisl substance are aggregates of ribosomes in neurons. If we want to see them, we can use normal synoptic staining, but to highlight it, we should use special staining. We can use Cressil violet or toluidine blue, and we have got a specimen where neurons of spinal cord are stained by toluidine blue. And yes, we have got also slides of blood, so-called blood smear and bone marrow smear. So smear stainings are used there, and it is the Pappenheim's methods. I will speak, or somebody who will have the lecture about the blood will speak about it later. Some pictures. Heidenhain staining is typical cytologic staining because it stains myofibrils or mitochondria. It's used mostly for muscle tissue, and it can show very nice striation of muscle if it is striated like cardiac or skeletal muscle. It is a photograph from our specimen. toluidine blue used to show Nissl substance in neurons, so you can see blue color of cytoplasm, and it isn't homogeneous blue color of cytoplasm, but some pieces of cytoplasm are more blue, darker blue, and it is Nissl substance. The same blue color is also in the nucleus, and maybe you know what is it. It is nucleolus, which is in fact of this composed from the same ribonucleic acid as ribosomes in the Nissl substance. Selective staining. Selective staining, as there was already written, are used for highlighting of some specific structures regardless of their location. Again, I would highlight these which are used in our box. You'll find there are several sections stained to visualize elastic fibers. Elastic fibers are special fibers of connective tissue, and they are usually invisible or hardly visible in synoptic staining because they don't bind basic or acidic dyes, but they can bind special dyes like orsane, which is brown, aldehyde fuchsine, which is purple, or black resorcine fuchsine if we use it, and such slides we have got. And then we have got a slide where mucus is stained by musicarmine, special dye which is specific for mucus, and mucus is red in this situation. Maybe you remember that mucus was declared as almost colorless in synoptic staining. It doesn't stain well. The mucus granules are white or very light, but if we use musicarmine, they are red, so you will see them very well in this one specimen. Neutral lipids, we haven't got a specimen where special lipid staining was used, but you should remember then that neutral lipids can be stained only in frozen sections, and the reason was already explained. Lipids are dissolved if we don't avoid lipid dissolvents. And then there are some another selective stainings used mostly in pathology. Again, there will be a short gallery of pictures. Orsane is one of elastic staining. It's brown, and so we can see brown elastic fibers here in the vessel wall. It is a wall of an artery, and we can see brown elastic fibers there. Next picture will show again wall of an artery, but elastic fibers are almost black because resorcine fuchsine was used. And the last picture from this series is another artery, little smaller magnification, and aldehyde fuchsine was used here, and so elastic fibers are dark purple or violet. You can see them here, violet fibers in the wall of this our artery. So yes, so this was elastic staining, and it is a special staining of mucus. Yes, we have got one slide in this staining as well. It isn't staining of mucus in our case, but staining of special extracellular matrix connective tissue. It is Alcian blue. Alcian blue is a special staining of acidic polysaccharides, acidic mucopolysaccharides, which can be found in mucus but also in connective tissue extracellular matrix. And Alcian blue is blue, of course, and so then mucus or this extracellular matrix is blue, so we can see here blue apical parts of goblet cells in the intestinal epithelium. You know this picture, but goblet cells were very light, almost white, never blue in the specimen you saw. Very similar thing, but it is this already declared mucicarmine. Mucicarmine is staining mucin red or purple. And carmine is staining glycogen and other similar neutral polysaccharides. Here liver is stained by best carmine, and we can see how much glycogen is possible to find in every hepatocyte. It wasn't visible it isn't visible in synoptic staining, but here we can see it. Lipids are stained in frozen section, where the structure is never so well preserved as in the chemically fixed and paraffin embedded tissue. Now, but the advantage of the freezing is that we can stain lipid droplets because they are not exhausted and extracted. So this red thing is a lipid droplet, which was white in the specimen you already saw last week in the practical training, and lipid staining can be also black. So here in this specimen there are black lipid droplets in adipose tissue, which you saw in paraffin section containing white bubbles. Special myelin staining, I think we can go on. Toluidine blue together with osmium tetroxide can stain myelin blue. Congo red is staining amyloid. It's a pathological protein, so it's used in pathology, and the same is the case of fibrin, which is also pathological. No, it isn't pathological product, but its staining is also used in pathology. Fibrin is normal physiological product of blood clotting, but blood clotting when found in the tissue is a pathological mark. Sorry, I skipped one picture, but I can go back and I'll show a slide about impregnations. Impregnations are stainings based on the silver or another metal reduction in the tissue. The procedure is quite complicated, and the result is very dark staining of some structures. It can be reticular fibers, which are extracellular structures, or it can be some intracellular material, mostly in neurons and glial cells, which are oligodendrocytes or astrocytes. The staining is here based on the affinity of cytoskeleton and silver salts. Then there are some another methods. Methods of staining are called according to scientists who introduced it. I would like you remember Gemery. Gemery, it was a Hungarian scientist, but he lived mostly in United States, and so you maybe can find his name with another pronunciation like Gemery and so on, but the original is Hungarian pronunciation Gemery, and his impregnation stains reticular fibers. There are special fibers of connective tissue. Then there are several neurological stainings, neuropathological stainings for glial cells or neurons. In our slide box you can find two impregnated specimens. One is a spleen where Gemery method was used and reticular fibers are black, and the photograph is here. So it is impregnation after gamma ray. These black network here are reticular fibers, and another impregnations for neurons and glial cells. You will see impregnation of neurons in the one specimen of brain. After staining, there has to be mounting, which are already described as the covering of specimen by very thin cover glass. Of course, this cover glass has to be sticked to the surface of the specimen by special mounting medium, which can be some resin or water-soluble medium, but the most frequently used is solacryl. Solacryl is a acrylic resin which is sticking the cover glass like glue, and the result is protection of our specimen. And when well done, the specimen which is covered by cover glass in solacryl and which is well dehydrated and well stained can be taken as a permanent specimen. It can be observed many times, it can be stored for many years, and if well stored in dark, it can be still used for new and new observation. In the electron microscopy, the situation is little different. Instead of staining, we are using contrasting here, and the specimen is never permanent. Specimen is only very short living. It is used only one time, usually in the electron microscope, and then it becomes useless. So we must photograph the specimens in the electron microscopy, because the photography is the only proof of our work here. So there aren't any permanent specimens. Contrasting is based on the affinity of some atoms and our tissue. These atoms should be heavy metals, because only they give contrast to the specimen. Normal light atoms like biogenic elements, oxygen, nitrogen, carbon, hydrogen, they are not giving contrast to the tissue, but heavy metals do it. We know which heavy metals are bound to the tissue, and so these metals we can use. Uranium and osmium were used during fixation and dehydration already. Now, during the proper contrasting, we can use uranium once more, but more concentrated solution of uranyl acetate, and we are using also lead as the citrate, but we can use also tungsten, we can use ruthenium, and again there are many contrasting methods. Contrasting agents then bind to proteins or phospholipids and make our picture recognizable and readable. Again, processors are in some laboratories for the contrasting, but we haven't got it, so in our laboratories everything done by hand. Grids are given on small droplets of contrasting solutions, and usually in dark, and it is contrasting. And the result is like this. You already saw many electron micrographs. So these parts of our specimen which bound the most atoms of heavy metals are black, like this granule, or heterochromatin, you know it, or collagen fibrils, which you don't know at this moment, but you will know them early. And these parts, which are black, are called electron dense, and the structures which don't bind the heavy metals are white, like this extracellular matrix here, or some pieces of nucleus, and they are called electron lucent in laboratory histological language. And there are some gray materials like a cytosol of this cell, which or this mitochondria, which can be declared as moderately or slightly electron dense only. Okay. I said at the beginning that there exists also scanning electron microscopy. SEM means scanning electron microscopy. What does it mean? It means that in this case the beam of electron doesn't go through the specimen, so we don't need thin section. The beam of electron is only moving along the surface of a specimen, and these electrons are reflecting, or they are creating so-called secondary electrons from the surface, which are detected, and we can see a three-dimensional picture of the surface of our specimen. So we don't need thin section. But our specimen has to be fixed and dehydrated, and not only simply dehydrated, but it has to be also dried, so no fluid can be in the specimen before we place it into the scanning electron microscope. So after dehydration, which is usually very similar to the dehydration for the transmission electron microscopy, there is a new special step, critical point drying, during which the specimen is dried by evaporation of liquid carbon dioxide under increased pressure. So we need special machine for this. And next step is mounting of specimen, and what is also important is shading of the surface. Shading of the surface of our specimen is necessary for the reflection of electrons. Without this, electrons can be absorbed by tissue, but we need to reflect them, or we need secondary electrons. So shading is in fact covering the surface by a very thin layer of heavy metal, usually gold is used here. So again, here in this step we need special tools. So the scanning electron microscopy needs a little special and different equipment of the laboratory. And this is the picture from the scanning electron microscope. It is inner surface of trachea, quite small magnification, so we can see many processes, mostly cilia, but also microvilli. You will know them after the lecture about epithelial tissue. It will be our next lecture. So you will know very early what do I mean by under the terms cilia and microvilli in the tracheal epithelium. And the last step of all histological methods must be observation, of course. It can be direct observation in the microscope, which you will do in the practical training, and which was done many years since the development of the light microscope, which was developed at the end of the 17th century. So the light microscopy is quite old method, and during the longest period of its use, there was the only possibility how to store the picture drawing. The microscopist observed the specimen and drew it to the paper. Quite newer thing is photography. It was discovered at the end of 19th century and used in microscopy during the first years of the 20th century. So it's approximately 100 years old, photography in microscopy. But it was very big improvement of the collection of pictures, because it was possible to capture the specimen and to get the picture without any long drawing. Now we have got digital photography, digital cameras, we have computers, special software, so we can do many things with the pictures from the microscopes, from light or electron microscope. We don't only simply photograph them, but we are analyzing them, reconstruct them three-dimensionally, and so on. To be able to do it, we must understand our microscopes. The light transmission, so-called bright field microscope, is in this picture, and you know it from our practical. You don't know it because you weren't here, but you will hopefully hear next week for the first time. And so you will see microscopes in our microscopic rooms. It is this type, and you will see that they have got 4 optic parts: light source, condenser, objective, usually more objectives which can be exchanged, and eyepieces. 2 eyepieces are here because our microscope are binocular. This is the picture. This thing in the corner is a picture of the first microscope which was developed by Antonio von Leeuwenhoek in the second half of the 17th century. It's hard to imagine how was it used, and how and what was the magnification, and what was the observation, what was the work with this microscope. But Leeuwenhoek personally was able to see cells. We know his drawings of sperms, for example, which are very small cells, so the magnification of his microscope was quite good. But of course the magnification and the picture, the correction of all aberrations is much better in modern microscope like this, our Olympus microscope. If we want, we can improve the contrast of our picture by using of special objectives, special condensers, special filters. So there are several methods how to improve contrast of light microscopy, mostly in situations that the contrast of the specimen isn't good naturally, like in microscoping of living cells, not in histology. Histology is a domain of the sections, of the stained sections, but cell biology and the research in the field of cell biology, it is based mostly on observation of living cells in light microscope. And living cells in bright field haven't got a very good contrast, but when we use variable improvements of the contrast, phase contrast, which is caused by a special condenser and objective, contrast is better. And differential interference contrast, the picture looks three-dimensionally. It is again special condenser used here. So it is possible to improve the contrast by a light transmission microscope. And there is also fluorescence microscopy. Maybe you know that there exists a physical phenomenon of fluorescence, which is the ability of some substances, which are called fluorochromes, to absorb light and to respond to this absorption by excitation of light by another and longer wavelength. And as the white light, which is produced by sun or many other sources of light, is composed of many colors which can be diffracted like in rainbow, the longer is wavelength of the light, the more red is its color, it's possible to say. The shortest wavelength is ultraviolet or violet light. The longest wavelength is or has the red line, and between is blue, green and yellow color. And so the color of the light of longer wavelength is different than the color of light with shorter wavelength, and this is the principle of fluorescence and fluorescence microscopy. Fluorescence microscopy is using white light, but it's filtered by excitation filters, so only one range of wavelengths is going on, usually not only one wavelength, but the spectrum of wavelengths is very reduced. This light, let's say blue light, is coming to our specimen, and if it is a fluorescent specimen, that means if it contains some fluorochromes, it excites light with longer wavelength, which can be, for example, green, and this green light goes back to the eyepiece through the barrier filter, where blue rays are filtered, where are blocked. Blue light can just reflect in this case, but it is blocked here, and so we can see only green light if it is produced by our specimen. So if this specimen has got some fluorochrome which is able to absorb blue light and excite green light, we can see it. It's the principle of fluorescence microscopy. There exist two types of fluorescence microscopes, transmission and epifluorescence. Much more used currently is the epifluorescent microscope, where the principle is in fact simpler, because we can use just one objective for illumination and observation together. Light is going through the exciting filter, and it is reflecting on the dichroic mirror, goes down to the specimen. Specimen is here. If it contains some fluorochromes. Emitted light is going back, but now it is going through the dichroic mirror. It doesn't reflect, and we can see it. We have got here again barrier filter, which filters some remaining blue light going through the dichroic mirror. So our picture is then clear, and so the so-called cube, which is situated in the epifluorescent microscope, every time has got special exciting and barrier filters and dichroic mirror, and there are cubes for the blue light and green fluorescency, and for green light and the red fluorescency, and so on and so on. There are many combinations. The fluorescent microscope was developing a long time, and there were many attempts to improve it, mostly to improve its focus. And one of these successful attempts was laser scanning confocal microscope, where also some Czech scientists participated in the development. And laser scanning microscope is a better epifluorescent microscope, which uses laser as a light source. It is one improvement, so not white light, but monocolor light from laser source is here used. And then the second improvement is a special confocal stop, where is very small pinhole, allowing to see only focused beams going from the specimen, and not unfocused beams which are filtered. And so detector here isn't eyepiece but special detector, because it's impossible to look at the light from laser, but detector is detecting only focused parts of our specimen. And next picture can show the difference of standard fluorescence microscope and laser scanning confocal microscope. It is almost the same specimen here, and you can see that the picture in laser scanning microscope is much sharper. But of course, laser scanning microscope is also much complicated and much more expensive, so not all departments can use it. The last several pictures are pictures of the electron microscope. This is transmission electron microscope. It was developed by Ernst Ruska in Germany in 30s of the 20th century, before the Second World War. Then their development was stopped during the war, so they were coming to the histological laboratories in early 50s usually. And so the method of electron microscopy is quite young, much younger than light microscopy. The first microscope was very primitive if you compare it with current microscope, which is here in our department. And the principle of the transmission electron microscope can be compared with the light transmission microscope. If you turn it up and down, the light source, or instead of light source, which is downstairs in the light microscope, is here electron gun, which is upstairs. Here is electron gun, which emits electrons. It emits beam of electrons which go to this column, where is the air evaporated or pumped out, so there is vacuum in this column, and a beam of electrons goes through several lenses, and their names are the same as you know from light microscopy. So there is condenser lens, objective lens, between them is a specimen. Of course, lenses are not glass lenses like in the light microscopy, but electromagnetic lenses. Specimen isn't on glass but on grid, which you know, and instead of eyepiece there is projector. So last lens is projector, not eyepiece, and the picture is projected. Picture is projected to fluorescent screen, which is here under this covering, and it can be also projected on photographic film, photographic emulsion, or in the most modern, and it is the case of our microscope, in most modern microscopes, to the digital camera. There exists special digital cameras with special chips which are sensitive for electrons and not for visible light. So the picture in this modern microscope is visible directly in the monitor. As I said, there exists also scanning electron microscope. I said something about it. It is working in two modes. It can use reflected or backscattered or secondary electrons. It has again electron gun, condenser, objective, but now there isn't specimen between condenser and objective, but there is a special beam deflector, and this beam deflector is causing movement of the beam along the surface of our specimen, which is under the objective lens. And this movement of the beam maps the surface of the specimen in a special raster, and the picture is then detected. The picture, that means this secondary or backscattered electrons are detected and visible on screen. So there isn't again any eyepiece, but picture is only electronic and it's only visible on screen. It is the photograph of a scanning electron microscope. Unfortunately, we haven't got it here, but you can see at least its picture, and this is the last picture of this lecture. So, thank you very much for your attention. I hope that this lecture can help you to understand how our specimens are made and what can you expect from them. Also, there were information necessary for the test you will write in 2 weeks, in fact, or 3 weeks during the 5th week, and there will be some technical questions as well. So thank you for your attention once more, and goodbye.